thank you for the and hi everyone. Sorry, there was a bit of a delay. My laptop did not correctly boot up. All right, so good to see all of you here. There seem to be quite a lot, 39 participants. That's quite a lot for a biostats workshop. So so before I actually start my workshop, I would like to know. So I would like this to be an interactive workshop. I would like you guys to respond to my prompts or my questions. And since this is going to be practical biostatistics workshop, I would like if all of you actually boot on your laptops or your PCs and start Jazz. I believe I had provided a link to Jazz in my um, description. So if anybody hasn't yet downloaded and installed Jazz, I will send the link in the chat
I've sent
box.
the link right now.
Yeah, yeah. So that one. So you can download Jazz. It's not a small file. It's a 700 MB file, I think. So but before I start, I definitely would like to know what your level in biostats is, or what your level in research is, or what you're interested in, so that I would be better able to conduct the session. And if needed, we could also have, I've made a small presentation to get you initialized on how to approach statistics. So if any one of you wants to put in the chat box or personal chat me, so are you so are you a beginner? Are you don't know anything about performing statistical data analysis, or um, are you a pro or intermediate? So okay, so you can either put it on the chat box, or you can unmute, or you can put it like personal message me, private message me. Um, all of that is fine. So okay, so I can see a few beginners here. So that is great. So I can start from the very basics. So I had prepared for that actually. I would have been surprised if most of you were um, experts. So that's great. Okay. Okay. So I think I have a rather elementary understanding of the demographic here. So all right, I will start with um, a, a very basic introduction to statistics and data and sample size calculation and so on and so forth. So I won't bore you, because this is going to be a practical biostatistics workshop. So I won't bore you much with the presentation. However, I would like you all to know that please bear through this presentation, because the concepts which I'll be talking about here will be very useful, since most of you have a relatively beginner idea of statistics. So I would like to start screen sharing.
What is your name again, sir?
So I hope all of you can see my screen. So first of all, I am Dr. Suhrud Panchawagh. I have just finished my medical school, and I'm currently a neuroscientist at the Center for Brain Research, Indian Institute of Science in Bangalore, which is the topmost institute in India for research. I am also the co-founder and CEO of Requestats, which is a startup company which enables, which is a consultancy service which enables new researchers to start out the research, and they provide help in consultancy. So I would also like all of you to take screenshots, because some slides which I have made in this presentation, I do not expect all of you to take down notes, which is very impractical. So I would encourage all of you to take screenshots of some of the slides which I am showing. So these are some of the resources. I have a YouTube channel. It's called StatStat. And in this YouTube channel, I actually aim to um, teach the principles of biostatistics and tutorials, which I'll be showing right now in JAS. So all of these tutorials are also present on my YouTube channel. So you can refer to my YouTube channel if you have any doubts, or if this session ends and you have any doubt, you can always go back to my YouTube channel and have a look. The rest of these resources are resources which are used in the top medical institutes or public health institutes in the world, Johns Hopkins, Harvard. So all of these books are excellent resources. So I would highly encourage all of you to at least go through these books if you have them in a library, or you can download PDFs as well. All right. So before I begin with some formal concepts, I would like to start off with how to calculate sample size. So all of you, I'm sure all of you know how important sample size calculation is um, for doing proper research. So your comparison test, your hypothesis test, all of those tests results are based on the validity of the sample size calculation. So ensuring that you have a perfectly valid sample size, you have an appropriate sample size is extremely important. So I will be discussing a few formulae. I don't expect this to be a full comprehensive overview of sample size calculation, but I would this would be an introduction to sample size calculation. Of course, there are more elegant methods vis-a-vis using softwares, which I will also discuss. So I would start off. So suppose we want to study. So we are aiming to conduct a study where we want to simply study characteristics of geriatric elderly patients with mild cognitive impairment. So this is a descriptive study. We are not looking to perform any hypothesis test, nothing, no fancy statistical analysis. We just want to study characteristics. We want to study your mean value, the mean values, medians, frequencies, percentages, just a very descriptive and exploratory study. So to calculate sample size, this is the Cochrane's formula. So I will, yeah. So you now take a screenshot of this slide, and basically, so this is a formula to calculate ideal sample size for a descriptive study or for a survey. Now, this sample size, mind you, is not a sample size for comparative studies. For example, if you want to compare two means, or if you want to compare two percentages or frequencies, this is not the sample size. I'll be discussing that in after a few slides. So what do all of these values mean in this formula, the Cochrane's formula? So Z is the confidence level. So that this is the measure of confidence in the precision of the result. So uh, this usually is, we typically take this Z value at a 95% confidence level as 1.96, or at a 99% confidence level as 2.58. So this value of Z is obtained from Z tables. So these are statistical tables. And for the level of confidence we are confident of in the precision of the result. So the so this is a very important statement. So the measure of confidence in the precision of the result. So how precise a result we have. So if we are 95% confident in the precision of the result, then your Z value would be 1.96. So for 95% confidence level, we have Z value of 1.96. For if we are 99% confident in the precision of our result, then the Z value from the Z table would be 2.58. So these are two values. These are, you find these values on the internet. So these are easy to find. If you just search up Z in sample size calculation, you'll get these values. Now, the P is the estimate proportion, or also the measure of variability. So unless we have a knowledge about the population we are studying, we leave it at 0.5, which means there's a maximum variability. Suppose we know the P value, the estimated proportion. Suppose a mild cognitive impairment, we have if we have a previous study which has been done, a previous epidemiological study which has been done, or if we have done a pilot study, or if an expert in the hospital or if an expert in an institution knows that the prevalence of mild cognitive impairment in this example is, say, 30% in the geriatric population we are studying above the age of 60 years, then the p-value would be 30% or 20%, whatever the proportion, estimated proportion is. So this p-value is we get this p-value from either past literature which has been published, or second, after performing a pilot study, or third, according to an expert's opinion. So if an expert has vast experience in this field and there is no prior literature, that we haven't performed a pilot study, then the expert's opinion or the expert's estimation of the estimated proportion is taken. Of course, 1 minus p is just the 1 minus p. So proportion here, mind you, is in decimals. So 60% would be 0.6, 30% would be 0.3. That's why we have taken 1 minus p here. Divided by e square. So e is the precision level. So this is the margin of error that we are prepared to tolerate. So suppose in previous literature, it has been suggested that the prevalence of the proportion of mild cognitive impairment is 30%. There has to be a certain error margin. So say 30% plus or minus 5%, 30 plus or minus 4%. So we cannot be completely 100% sure, right, that the proportion is actually the true proportion of the population. So E is the precision level of the population. This can either, if it is not provided in previous literature, this can be arbitrarily assumed. So plus or minus. So if there is no prior knowledge, then usually what is done is the precision level is set at 4% to 6%. So it varies from 4 to 6. All right. So if you have taken a screenshot, I will move to the next slide. So this is what I was talking about. So based on a relatively recent study in India by Mohan et al. This is a study which I've cited here. The prevalence of mild cognitive impairment, or MCI, in adults older than 60 years is found to be 26%. And the confidence limits are from 22% to 30%. So we can see that 26% plus or minus 4%. So the precision level would be plus or minus 4% here. And the P would be 26%. So plugging these values into the formula, confidence formula, we have at a 95% confidence level, which would be correspond to a Z value of 1.96. After plugging in all these values into the formula, would get a sample size N of 462. So the ideal sample size to study patient characteristics, a survey or a descriptive study in this population, would be 462, according to the parameters we have taken. Mind you that if the precision level ranges from, say, 20% to 32%, that would be plus or minus 5. That would mean an E is plus or minus 5. Your sample size would decrease because the precision is now lower. So you are less sure that your true population proportion is 26%. So your sample size will also decrease if your precision level is precision error of margin is higher, but the precision is lower. All right, so I hope all of you have understood this. If anyone has not understood, you can unmute yourself again, put something in the chat box. So due to constraint of time, I will move forward without stopping too much here. At the end of the session, you can always ask me any doubts you have. So this was a sample size calculation for a descriptive study. Now, this one, I will talk about calculating sample size to compare between two proportions, to compare between two frequencies or percentages. So I think all of you in neuroscience are from biology background, and all of you have a fair bit of understanding about medicine and the human body, except for the aside from the brain. So this actually presentation was catered to medical professionals, but I will explain if something is not clear. So we want to perform. So suppose we want to perform a study where we want to compare healing rates of wounds in diabetic patients. So I'm sure most of you know that diabetes mellitus predisposes patients to delayed wound healing due to a number of mechanisms. So in this study, we want to compare healing rates of wounds in diabetic patients who have been treated using one of two methods. So these methods are, the first method is regular dressings. So dressings are basically protection. So it's a covering of the wound, which helps the wound to heal and it is not exposed to the outside environment. So this is the first. So one group of patients is only being treated with the regular dressings. And now the second group of patients, there is a new super healer ointment in town. The new super healer has been released. It is FDA approved and everything. So we want to study whether the application of this new super healer actually increases or improves healing rates, or whether there's a difference in healing rates between both of these methods, or whether both of these methods are the healing rates are not different in both of these methods. So the super healer is not truly effective. So we want to study this, whether there's a difference in both of these methods. So before actually going into calculating sample size, this table is a very useful table to get an idea of the outcomes of a study of a statistical data analysis we have performed a study. So I encourage all of you to take a screenshot of the slide. So I'll explain what this table means. So the vertical columns stand for the reality or ground truth. So what exists in reality, or what is the ground truth? So the first column. So if a difference exists, the first column says that a difference truly exists. That is, in reality, there is a difference, say, between these two types of treatment. So there is a difference between application of the super healer versus just regular plain or dressing changes. And the right hand side column indicates that in reality, no difference exists. Ground truth is that there is no difference between the new super healer and your regular dressing genes. The rows indicate the results of your statistical test. So suppose you collect data and you analyze data and you perform a statistical test. The first row indicates that your statistical test has found a significant difference, or your p-value is less than 0.05. So your first row signifies that. Your second row, the bottommost row, signifies that your test has not found a significant difference between these two treatments. So now the consider the 2x2 table on the bottom right screen. The top left cell indicates, you can see that in reality, there is a difference between these two treatments, and your statistical test, your study has found a difference. So the H0 is and the H1 are just denotations of your null and alternative hypotheses respectively. I will briefly touch upon what these mean in a few minutes. So basically, when in reality, there is a difference between these two types of treatment, and your study also finds that yes, there is a statistically significant difference. This is obviously what we mean by a true positive. So this is truly positive. And this is what we mean by power. Now I'll come to you. Most of you might have heard this term. I'll come to what this means exactly in a bit. The top right cell is a false positive. So in reality, the ground truth is that there is no difference. No difference exists, but our study finds a statistically significant difference. Our study says that yes, there is a difference, but in reality, there's no difference. So that is, of course, a false positive, also denoted by alpha, also known as a type 1 error. false positive, alpha, type 1 error. All right. The bottom left cell. So in reality, there is a difference between the regular dressing changes and your super healer, but your study fails to find a significant difference. Your study does not actually find that there is a difference. That is, of course, a false negative. So it is your study is falsely showing that the difference is negative. This is also denoted by beta. So this is also known as a beta error. This is known as alpha error. Beta error is also known as type 2 error. Alpha error is known as a type 1 error. So alpha, beta, 1, 2. So you can remember it like that. And the bottom right cell, of course, is a true negative. So in reality, there is no difference, and your study also finds no difference. That is great. That's a true negative. So that is also desirable. All right. So arbitrarily, usually while calculating sample size, you set a power that is 1 minus beta or true positive. You set your probability that you'll get a true positive to 80%. So this is an arbitrary thing. You set the power at 80%. You can also set it at 95%, 99%. So anything above 80% is fine. Anything below 80% is not fine. That translates to 20%. You are willing to accept that you will get a false negative. So if you set your power at 80%, you accept that you have a 20% chance of getting a false negative. If you set your power at 95%, you accept that there's a 5% chance of obtaining false negatives. So as you increase your power or you decrease your probability of getting false negatives, your sample size will also increase. So remember this. So as you increase your power, you are decreasing the probability of getting false negatives. And when you plug this power level into your sample size formula, your sample size will increase. So higher power, higher sample size needed to prove that. Your significance level is also known as alpha or type 1 error. So this is your significance level, and arbitrarily we set it at 5%. 5% is also 0.05. So where have you heard 0.05? It is your threshold of your p-value. So when you say that you have set a significance level at 5%, that means you are ready to accept 5% false positives. So you're ready to accept 5. So anything above 5% is not desirable, and anything lower than 5% is all right. Note also that if you decrease your false positive rate or you decrease your significance level to say 2% or 1%, your sample size will increase. If you increase your power, sample size increases. Or if you decrease your significance level, your sample size increases. This is important. And this table is a very helpful table to understand this concept. All right. So suppose we know. So we know that regular dressings have healing rates of 60%. So there exists prior literature, there exists a common consensus that just regular dressing changes in diabetic patients would lead to wound healing rates of 60%. So 60% of patients will get, who have wounds will get fully cured or healed. And now we are expecting this new super healer to improve healing rates to 80%. So there exist small pilot trials, there exist small studies, there exist animal studies which suggest that this new super healer improves your wound healing rates in diabetic patients to 80% from 60%. Right. So this, you can take a screenshot of the slide. This is the sample size required in each group. Note that this is for each individual group. So the super healer and the regular dressing changes group only. So you might say, oh, Suhrud, you talked about significance level and power and how you need to plug it into the formula. Why aren't we seeing this here? Because this formula already takes into consideration that you are assuming a significance level of 5% and you are assuming a power at 80%. So keeping a power at 80% and keeping a significance level at 5%, this is the sample size calculation formula that is needed to test a difference between these two groups of proportions. So this is sample size for a difference in proportions. So here V and W denote your healing rates or the proportions, what you're expecting, or whether priority suggests or there's a pilot study, right? Okay. So plugging your values into this formula, we get after calculations, we get a samples of 80. Note that this is for each group. So you will need 80 patients in your regular dressing groups, and you will need 80 patients in your um, super healer group. So total of 160 patients. All right. Now the last type of samples as calculation formula is samples as calculation formula to compare two means. So to test the difference of means in two groups. So the first one was for a descriptive study, there was no comparison. The second one was for a comparison between proportions, and this third one is for a comparison between means. Okay. So suppose, so total cholesterol is a biomarker for lipid levels in the blood. So you have lots of parameters: total cholesterol, low density lipoprotein, high density lipoprotein, very low density lipoprotein. So all of these are your lipid parameters which you routinely measure in patients. So one such parameter is your total cholesterol levels in your blood. So suppose we want to compare, and these total cholesterol levels are in a range. So from 0 to 200 to 300 or whatever. So usually the cutoff range for high cholesterol is 200. So if you have total cholesterol of more than 200, that means you have hypercholesterolemia. If you have less than 200, it means that you have normal cholesterol levels. So suppose we want to compare the mean total cholesterol levels in patients in two groups. Now, what are these two groups? So one group of patients have been suggested only dietary changes. So group one is only being recommended dietary changes. So have a low lipid diet, don't eat fried foods, don't eat fast foods, have a low salt diet, have a diet rich in fiber, in green leafy vegetables, rich in protein, meat, eggs, so on and so forth. So this is one group. And the second group is consists of the patients who have been given statins. Now, statins are what are known as lipid lowering agents. So these actually lower your cholesterol levels. So these are drugs which are given in patients. These are two groups. So one group who have been prescribed statins and one group who have been prescribed only dietary changes. So we want to compare the mean cholesterol levels in both of these groups. So this, you can take a screenshot of the slide. This is the sample size calculation formula that you would use to check your cholesterol levels. So this again assumes that your significance level is set at 5% and the power is set at 80%. So that's why you don't see any variables for these values. So again, your Z is the same Z which you had seen in your first slide. Okay, so again, for a 95 at a 95% confidence level, you will have a Z value of 1.96. At a 99% confidence level, you will have a Z value of 2.58. The SD denotes your pooled standard deviation. So all of you might know that the standard deviation is a measure of dispersion. So how dispersed are your data? So standard deviation is a measure of your data dispersion, and your standard. Okay. So how there's a chat. How would we know for a certain condition we are supposed to use which formula? Right. So it depends on the type of data. So if you are measuring numerical data or continuous data, you would use this formula to compare between two means. If you are measuring, suppose, frequencies, so you are measuring outcomes or proportions, you would use the second formula. If you are performing a descriptive study, you would use the first formula. So I hope this is clear. If it isn't, you can put a chat, put a comment. Um, Okay. Okay. So your SD stands for your pooled standard deviation. That is your standard deviation from both of these groups. So you pool all of the patients from group 1 and group 2 together, and you find out the dispersion of their cholesterol, total cholesterol levels. So your SD stands for your pooled standard deviation, and your E, you guessed it, is the same E as the first formula. It is the desired margin of error for difference in means. So I'll explain what this desired margin of error for difference in means. So suppose in the group treated with statins, your total cholesterol levels have dropped. So higher cholesterol levels means it's a bad thing, and lower cholesterol levels obviously means it's a good thing. So suppose in group 2 who have been prescribed statins, their mean total cholesterol levels are say 170, and in the group who have been prescribed only dietary changes, no drug intervention, no statins, suppose their mean cholesterol levels are 190. So the difference in 170 and 190 is of course 20. So how sure are we, what is the precision of this measurement? So how sure are we that it is 20? So this will also, of course, reflect, we would also of course have to report a margin of error for this difference in mean. So 20 plus or minus what? So are we sure that 20, there's a difference in 20 plus or minus 5, 20 plus or minus 4? So what is actually, so this is known as confidence interval. So this margin of error is what is denoted by E. So 20 plus or minus what? Okay. Now moving forward. Suppose suppose we have performed a pilot study now in 20 participants. So 10 each. So 10 in the dietary changes group and 10 in the statins group. So based on this pilot study, we know that the standard deviation, that is a dispersion of total cholesterol in both of these groups, so pooled SD, is 20. And now we want the margin of error to be no more than 6. So the milligram per deciliter is your unit of your total cholesterol measurements. So we want the margin of error to be no more than 6 milligram per deciliter. That means we don't want the margin of error for difference in means to be more than 6. So plugging these values into the formula, your Z would of course be 1.96 for a 95% confidence level. Your pooled standard deviation from our pilot study was 20, and the precision level for your difference in means is 6 plus or minus 6. So plugging all of these into the formula, we get an N of 86 in each group. So 86 in your statins group and 86 in your dietary changes only group. So total of 172 participants or patients in your study. So this was all about sample size calculation. I hope you have taken screenshot of the slide and formulae. If you haven't, you can let me know. I would go back to the slides. So now I will run through the remainder of the presentation, but because I am sure that all of you are itching to get your hands dirty with data. So if you haven't yet installed JAS, please install it right now, download and install it right now, because it is crucial for you to take practically do statistical data analysis with me. If you don't have the link, please put it in the chat box. Someone can share the link again. All right. Now moving forward. This might seem elementary to some of you, and this might be something new for some of you. So I will go through it anyway. So types of data and variables. So the first type of data. So why do we need to know types of data and variables? So we need to know this because the type of statistical test which we are going to use depends on what type of data we have, whether we have continuous data, whether we have numerical data, whether we have categorical data. So so your type of statistical test, your choice of statistical test depends on what type of data you have. That is why it is important to know what types of data there are. The first type is quantitative data or numerical data. Very easy to understand. These are numbers. So in our previous example, your total cholesterol levels, which range from 0 to 400, 300, 400, are your numbers or numerical data or quantitative data. This is also known as continuous data. So continuous data, quantitative data, numerical data. So a special type of numerical data uh, is ordinal data. So ordinal, so you can glean ordinal ordered. So ordered data from suppose lowest to highest. So from 0, 1, 2, 3, 4, 5. So all of these are ordered data. So since all of you are from a neuroscience background, most of you might know your CDR. So your CDR score for dementia, your clinical dementia rating score. So this is of course from 0 indicating normal healthy patients, 0.5 indicating mild cognitive impairment, 1, 2, 3, so on and so forth indicating dementia. So this is an example of numerical data numbers, but these are ordered. So 0 indicates that these are healthy patients, they have no dementia, no cognitive impairment. 0.5 indicates that they have MCI, mild cognitive impairment. 1 and onwards indicate that they have dementia. So this is an example of ordinal data. So this is a special type of continuous. Continuous data is expressed in terms of numbers. I would encourage all of you to now start using the terms continuous and ordinal instead of just quantitative or numerical, because this is important to differentiate between continuous data and ordinal data. So continuous data is expressed in terms of numbers. For example, say blood pressure readings, 120, so systolic blood pressure readings, 120, 144, 152, 96, 180. So these are numbers. So this is continuous data. And there is a certain classification for your blood pressure readings. It is known as the American Heart Association classification. So AHA stage 1 means it's normal. Elevated, say 2. Stage 1 hypertension is 3. Stage 2 hypertension is 4, and hypertensive crisis is 5. So if you number all of these are from this is an ascending, so from lowest to highest. So 1 indicating normal blood pressure and 5 indicating very high blood pressure. So this type of these are numerical data numbers, but these are ordered from lowest to highest. So this is a type of ordinal data. The second type of data is qualitative data, also known as categorical data, also known as nominal data. So nominal names, categories, categorical, qualitative data. So these would give you an idea of what type of data. So these are not numbers. These are certain attributes of the population or certain characteristics of the population. All right. Okay, so we have 15 minutes to go. So 15 minutes to the workshop ends. I'm not sure.
Yes.
Oh, that's too less. I was told that it was going to be for 2 hours.
Oh, we have another workshop in 15 minutes.
Oh, okay. So in that case, what I would encourage all of you to do, because it is not going to be feasible to complete everything. So, okay. So I would encourage all of you to open JASP. So I will introduce you to the software and we can do some data analysis in JASP. Okay. So can participants share their screens? So
sorry, we have much more time than that. I apologize. We have till 1 p.m. ET, so 10:30 p.m. your time.
yeah, so I think we have around 1.5 more hours.
Yes, correct.
Yeah. Okay, great. So that was what. Okay. All right. So I'll run through presentation. So the second type of data. So first was numerical data, quantitative data, continuous data. The second type is qualitative data or categorical data. So this data represents like certain attributes of a population, say names or labels. So suppose we have, so take the example of your total cholesterol levels. So we had two groups. So one group was your statins and one group was your dietary changes. So your statins or dietary changes, so this variable, the treatment offered, is categorical data. This is not numbers, this is a certain attribute of the population or labels given to categories. Uh, so this example also, suppose we have three groups of patients, one group taking ACE inhibitors. So ACE inhibitors and angiotensin receptor blockers, ARBs. So these are types of drugs which improve your blood pressure or decrease your blood pressure. So this category or the type of treatment given. So this would be an example of your categorical data. So I have explained this. I would not want to spend much time. All right. So what is a dependent and independent variable? This is very important. Please pay attention here. So dependent variables are variables which are outcome variables. So these are your variables which you are measuring. So these are response variables which are being measured or tested in a study. So for example, in our previous example, your total cholesterol levels, which we are measuring, were the dependent variable. So that was what we are measuring. And the independent variable are variables which affect your dependent variable. So in that case, type of treatment given was your independent variable. So either statins or dietary changes. So that was your independent variables. So independent variables are variables which affect your dependent variable, and your dependent variables are the variables which you are measuring or testing in your study. All right. So since I've explained this, I won't want to spend more time. So this is what I was leading to. What statistical test to use is dependent on what type of data you have. Before running any formal statistical hypothesis test, we would want to explore the data. We would want to explore, describe our data in terms of means, medians, standard deviations, interquartile ranges, frequencies. All of that I will show how to do in JAS in just 2 or 3 minutes. Let me just finish the presentation. So we want to do explain data analysis first. We want to visualize our data first, and you can now take a screenshot of this slide because this slide explains the tests you will use depending on the type of data you have. So suppose your dependent variable or your outcome variable, the variable which you are measuring, is numerical or continuous, and your independent variable is categorical or qualitative. So in the example of total cholesterol, so total cholesterol was a dependent variable, it's a continuous numerical variable, and the type of treatment given, statins or dietary changes, was your independent variable or categorical variable, categories. In this case, we would use a t-test if we have two groups, and analysis of variance if we have more than two groups, or linear regression if we want to build a model which is flexible enough. If our independent variable was also continuous or numerical, then we would have had to use a correlation technique or a linear regression technique. Now, if our dependent variable was black and white, like categorical, yes or no, or in two categories or more than two categories, so that is the outcome variable which are measured was in two categories. So if it was, say, take the second example. So healing and non-healing, yes and no. So wound was healed, yes, not healed, no. That would be a categorical dependent variable. And if our variable which was affecting this wound healing, so in the first case, the type of treatment, that is regular dressings or a super healer, that is also categorical. So a type of treatment, we would use a chi-square test or a Fisher's exact test. So both are used. So I'll explain the indications in the software when we are performing the analysis, or we would use a logistic regression technique. And if your independent variable in this case was numerical or continuous, would use a Lursky regression technique. All right. So I hope everyone has taken a screenshot, and I would encourage that after taking a screenshot, just open the screenshot and keep it somewhere which you can refer to when we perform our analysis in class. So just keep it handy, the screenshot. All right. Now, before performing any type of statistical test, there are certain assumptions of a statistical test that we need to fulfill or satisfy. So I would explain those while performing tests. Parametric, non-parametric, I would explain while in the software itself. Okay. Okay. So null and alternative hypothesis. So the null hypothesis is the hypothesis of no difference or statement of no difference between certain characteristics of population. So in the first example, in the second example, between regular dressings and the super healer, your null hypothesis would be that there exists no difference in the healing rates of wounds between these two groups. So group given regular dressing changes only and group given your super healer. The statement of no difference. And alternative hypothesis is a statement that yes, there is a difference between these two groups. Just so you know, alternative hypothesis can be one-tailed or two-tailed. So two-tailed means that we don't want to comment where the difference is. So we don't want to comment whether super healer is better or the regular dressings are better. So we don't want to comment that. We just want to see that there exists a difference. We don't know the direction of the difference. And one-tailed alternative hypothesis means that we are hypothesizing that there is a direction. So we are hypothesizing that the regular dressings are better than the super healer, or we are hypothesizing that the super healer is better than the regular dressing. So that is one-tailed. Two-tailed means we are not setting a direction to the alternative hypothesis. That we are just blindly seeing that there is a difference. We don't know where the difference is or what the difference is. Okay. Okay, so I would briefly, in 1 minute, I would want to touch upon what the p-value is. So we have now planned the study. We have formulated our null and alternative hypotheses. We cannot expect that we would be correct about the outcome of the study 100% of the times. We have to settle for some level of confidence or the significance level for which we want to be correct. So we have to settle for some false positives. We can't be correct 100% of the time. So this is what we mean by alpha. If you remember the alpha or the type 1 error or the significance level, so this is alpha is the probability that we are going to be incorrect. There are also false positives, if you remember. Arbitrarily, it is, so as I've said, significance level is set at 5%. So we want to be correct about an outcome 95% of the time, or we are willing to be incorrect 5% of the times. So that is your alpha is set at 5%. So this is arbitrary, mind you. Now we perform the study. After planning the study in excruciating detail, we analyze our data, we collect data, we analyze data statistically, and now we get a p-value from our statistical test, t-test, correlation, chi-square test, all the tests you have seen on the previous slide. So this p-value sometimes also denoted by PR. So some softwares or some literature might give a PR instead of the p-value, or might also give you a capital P value. So this is the probability. So probability, and otherwise said p-value less than 0.05, we reject the null hypothesis. So this how have we arrived to this 0.05 magical number? By setting a significance level of 5%. That is, we accept a 5% chance that we are incorrect about our result. Very formally stating, p-value is the probability of obtaining test results at least as extreme as those observed during the test. This is a very formal way of stating the p-value, and simply put, this means that less than 5% of the results occur due to chance. So the statement above is a formal formal definition of the p-value, and the statement, the last statement, is your informal statement, which is easy to understand. So by setting a p-value of 5% or 0.05, we are saying that we accept that there is 5% or less than 5% chance that the results occur just due to chance and not actually present. So false positives. So this is, I won't go into the details of all of these. I would now encourage all of you to start boot up JAS. I will do the same. If anyone has a doubt, please put it in the comments or please unmute. So how I want this to proceed is I would explain a couple of examples of how to perform statistical analyses in JAS. And after that, I would so I would ask for if there are any volunteers, and they could probably share their screens, and I would guide them on how to what they are doing and how. So I want this to be an interactive session. These sessions are best done offline because it is easier to do so. But since we don't have any option, we would settle for this, which is almost as good. All right, so I hope all of you are seeing this screen, and this would be sort of like a tutorial. So if you can just minimize JASP into a small screen and screen sort of thing, and can keep that tab in any corner. You can follow what I'm doing, and you can perform data analysis in JAS. So does anybody have a problem loading JAS or downloading JAS? I'll take some time, some 10-15 seconds before I start. So you can let me know in the chat box or unmute and let me know if you are facing a difficulty. So I want to know who all. I need a minute. Okay, perfect. So I want to know who all haven't downloaded JAS or do not have access to JAS. So in that case, I would go a little slowly and explain. So you can direct message me or yeah, just let me know if you don't have JAS or not downloading JAS. In that case, modify that. Okay, so before I start, let me introduce you to the JAS interface. So this is JAS. I'm sure all of you have heard about SPSS, the statistical package for the social sciences. So it had been taken over by IBM. It's a paid software. Unless you have an institutional access, I'm guessing an individual license might cost a bomb. So so that is, if you're an individual researcher, it might not be feasible to buy that software. The second, another good software is Stata. Stata is excellent, and I think for individual licenses, perpetual licenses, it is relatively reasonably priced, but it has a learning curve. Third good software is R. Now, R is a programming language, so you would need to learn how to code in R. Or if you have prior knowledge of, say, Python or any other C++ or any other SQL or some programming language, then it might be easier for you to pick it up. I would say among all the softwares for data analysis, R is the best software because it's open source, and because of it being open source, people are contributing to R, people are making new packages, and the functionality of R is heavily dependent on their packages. So so I would say R is the best. Second would be probably Stata, and third would be SPSS. JAS runs on R in the backend, so JAS is equally as good as R. JASP is free, so that is a perk. And if so this is a quite a recently developed software, and people have questioned the validity of the software and the results. So R actually JASP actually runs R in the backend. So R, you can think of JASP as a nice, pretty graphical user interface for R. So all of the analysis done in JASP, in the backend, the R code is going on. So it is a totally valid software and completely credible. All right. So I hope Sarah has booted up JASP. If anybody hasn't, there's still time. I'll just be explaining descriptive statistics and not any type of statistical test right now. So you can, take that time to load up JASP. All right. So you might be wondering why I haven't shared any data set. That's because JASP already has sample data sets which you can work on, and these are included in the software itself. So you don't need to download any other data set, or don't need to have any other data set in hand to perform analyses. So that's why I haven't shared any data set. So how can you access this data library? So you might be seeing fewer modules than what I have. So just if you want more modules, you can just select this plus icon, which will show you the modules menu. And here you can select or deselect whatever module you want if you want something extra. I would personally suggest that you go for equivalence tests because those are pretty commonly needed. Meta-analysis if you are performing meta-analysis, and power if you are performing power analysis, sample size calculations, and so on and so forth. So these three modules are pretty useful. All right. So to load an example, okay. Would the PowerPoint slides be shared? Yeah, sure. I can share that. I had sent it to the Simply Neuroscience team. You can share it forward. So I'll share the PowerPoint slides. The PowerPoint slides, most of the content can also be found on my YouTube channel. So the stat. This is my YouTube channel name, and most of the content is covered in the YouTube channel. So there's a playlist, the core concepts playlist is covered in that. I would share the slide, of course, no problem with that. All right. So just click on these three horizontally stacked sort of bars, which will go to your show main menu. And here go to open, go to data library. And here you have lots of folders which contain all your data sets. So just to repeat myself, if anyone has not followed me, these three stacked rows show main menu in the top left corner of your screen, three horizontal rows. Then after that, the open option. After that, go to the data library option, the last data library option. And here you will see all of these folders. If anyone has a problem, just please unmute yourself and let me know, or put it in the chat box so I'll know if anyone has a problem. So first, as I've already mentioned, before performing any type of formal statistical test, we would like to perform descriptive analysis or summary statistics or exploratory data analysis. So I would encourage all of you to open this descriptives folder, which contains this sleep data. Now, if you hover around these two icons, if you hover around the green JASP icon, you would see that it is telling you open JASP file. So by clicking this, you would open a JASP file which contains data. So all the tests which have already been performed by the author, and you would get a description of the data set and everything. So this essentially contains your refined data analysis with all the explanations. Because this is a workshop, I don't want you to open this JAS file. Please leave it after the workshop when you would be exploring the software yourself. So you can open JAS file and see analysis. If you see the icon, the sort of table icon to the right of the green icon, it says open data file. So it would just contain your plain data set and no analysis which has been done beforehand and no explanation. So I would be explaining the data set and everything. So please open the data file and not the JAS file. If you have accidentally opened a JAS file, it's fine, no problem. But I would encourage you all to open the data file, not the JAS file. All right. So I'd like to explain this data set first. So you can see that you have 20 observations, which you can understand from the leftmost column. So 1 to 20. And we have, it seems we have 3 variables: ID, group, and extra. Now, what are these small icons? What do these mean? So I've already explained the types of data: continuous, ordinal, and nominal or categorical. So scale means that you have numerical data or continuous data. So if you just click on the small icon, you'll see what these data mean. So this scale, small scale or ruler, stands for continuous data. This stacked bars from lowest to highest ordered, this is your ordinal data or ordered data. And this small like Venn diagram sort of icon, this is your nominal data or categorical data. So JAS automatically detects and defines what type of data you have. If you are not in agreement with the type of data, you can manually select the type of data here. So if you think that this should be a numerical continuous data, you can select that this is scale data. If you think that this should be ordinal, you can select ordinal. If you think this is a nominal data, names or categories, select categorical data. This is of course a nominal data, the patient numbers. So this would be a nominal type of data. All right. So what does ID mean? So ID of course means the identity, the ID number. So it seems that there are 10 patients, and these patients, so there seems to be 2 groups. So the middle column denotes group. So group 1 and group 2. So what this study is that these 10 patients initially have been given one type of sedative drug, and after for 1 week they are being given some sedative drug for 1 week daily, and after being given that drug they have left a 1 week washout period where they have been given no drugs, and in the 3rd week after the washout period they are being given drug number 2, so a second type of sedative drug. So in the 3rd week, the same 10 group of patients. So suppose group 1 say that they have been given a type of benzodiazepine known as lorazepam, and group 2 is a second type of benzodiazepine or sedative, say diazepam. So lorazepam and diazepam both are sedative drugs. They put the patient to sleep, they make you drowsy, and so these are sedative drugs. So in the 1st week, patients have been given lorazepam. 2nd week is washout period, no drug, and 3rd week diazepam, drug number 3. The first column, extra, denotes the number of extra hours, average number of extras the patients have slept in the week. So if you see this 0.7, so patient number 1 or participant number 1 has slept 0.7 extra hours in the 1st week on average. The second participant has slept lesser than average, lesser than he would normally sleep. So lesser by -1.6. hours, so on and so forth. So the leftmost column is your the number of extra hours slept. Negative values means that on average they have been sleeping less on the drug. Positive values means they have been sleeping on average more than normal. So before running any type of comparative test, so whether drug 1 is better or drug 2 is better in terms of increased sleep, we would want to perform some descriptive analysis. We want to explore the data. So how the data is. We want to explore how many percentage of patients are being given group 1, group 2, what their mean ranges, what their mean values are, what their medians are. So the first module, if you see this descriptives, this will give you your summary statistics or exploratory data analysis. So this will give you your measures and tendency, your means, medians, modes, your measures of dispersion, standard deviation, interquartile range, your frequencies, percentages, so on and so forth. So just click on this module and it will take you to descriptive statistics. So let's wait for the initialization of the module. My laptop unfortunately has lower RAM, so it takes some time. So if any one of you is unsure about what we are doing, please don't hesitate to unmute or leave it in the chat box. So I have not yet started the interactive part of the session. I mean, I hope that if anyone has a doubt, it will be interactive. But after this analysis and the one more analysis, I would want somebody to share their screen, their JAS screen, and I would like to guide you through it. Okay. So this is the JASP interface. On the left side of the screen, you have these are the options. So you would tinker around with these options to customize your analysis and what type of analysis you want. The right part of the screen is where your results, your tables, graphs, everything will be situated. So after you have given some options and customized your analysis. So if you hover around these variables, you can actually select 1, 2, 3 like this. If you want to select more than one, can hold down the control button for Windows. I'm guessing the command for Mac users, and select more than one variable at a time. So you can, so if you want, so put, okay, chat box. Okay. All right. So if you want, you can drag and drop these variables into this variable box, like drag and drop. Or if you select this variable, you can just click on this arrow button, the right side arrow, and you can drag and drop the variable here. Okay. So when you put your extra number of hours in this variable column into your variables box, you will automatically get a table. So these are predefined settings that JASP is already set, and you can customize these. So basically, if you want if you want this to be in a horizontal manner, you just select transpose descriptive table, and it will give this to you in a horizontal manner. So if you have lots of variables which are here, then a horizontal table might make more sense. So it's just an aesthetic preference or practical preference. There's no such, it's up to you. So you can see that you will have 4 sort of drop down menus: statistics, basic plots, customizable plots, and tables. So if you drop down into statistics, you'll see that you have lots of options to customize your table, your descriptive statistics. So by default, JASP selects valid, missing, mean, standard deviation, minimum, maximum. So valid denotes the number of valid data points you have. Missing denotes if you have any missing data. Here in this case, we don't have any missing data. That's why we have 0. So you can just deselect this. Sometimes it is better to report that you have some missing data. So you can select this missing data option and it will just give that to you. So suppose we want medians or modes, it's as easy as just selecting this checkbox. If you want the interquartile range, if you select quartiles, it will give you 25th, 50th percentile, 75th. So 50th, of course, is the same as the median. So that's the same. And you'll get an interquartile range, 25th to 75th percentile. The Shapiro-Wilk test, we'll come to that in a bit. The skewness and kurtosis both are measures of how your data is distributed. All right. So I won't dwell on other points. If you want customized percentiles, then you can just select this percentiles checkbox and suppose, let's say I want the 33rd and 66th percentile. So I would write these down, and if you see here, we have these customized percentile values. So just put a space between the two values you want, and you'll get those values. Right. So the second thing I would want to discuss for a continuous variable or a numerical variable is after after seeing your measure of center tendency, that is your mean and median, your measures of dispersion, that is your standard deviation, minimum, maximum, and interquartile range, I would now want to visualize my data. So how do you visualize continuous data? You can plot histograms by going into basic plots. Just select the drop down option, basic plots, and select distribution plots. It will provide a histogram like so. And if you want a density curve, just select this display density checkbox. It will give you your distribution curves. Now, let's not lose sight of the fact that we have two groups. So group 1 being given lorazepam and group 2 being given diazepam. So there are two groups of sedatives. So if you remember, so we don't want to see the data as a whole. We want to group between lorazepam and diazepam. We want to see whether there's a difference in the number of X-stars slept between these two groups. So what you can do is that just select this group variable and just drag and drop this into this split box. Once you do that, this table would automatically split into group 1 and group 2. So prima facie, we can see that the average number of X-stars slept was higher in group 2. So those being given diazepam slept higher than those being given lorazepam. So the same patients, when they were given diazepam, slept more than when they were being given lorazepam. So we can see even the median is higher. Standard deviation or the dispersion approximately seems to be the same between these two groups, which is actually a good thing. And your interquartile range, you can see that it is higher in your group 2 or your diazepam. And even in your histogram and density plot, you can see these two density plots stacked against each other. A second type of plot, which I would like to show, is a box plot. Now, box plots are visual representations of your medians and interquartile ranges. So just to reiterate, your median is your 50th percentile. So your median is the value which divides your data into 50% to 50% data points. Your interquartile range ranges from the 25th percentile to the 75th percentile. So 25th percentile means that there are 25% of values which are lower that data point, and 75th percentile means that there are 25% values which are above that data point. So that is your interquartile range. So box plots are visual representations of your median and interquartile range. So how can you get box plots? Just select this third customizable plots drop down option, and you will see an option for a checkbox, box plots. So when you select that, let it run. Let it render the plot, and you will get your visual representation like so. So immediately we are very nicely able to see that in group 2, the median was also higher and the interquartile range was also higher. So we can, visually see that in group 2, the participants have slept an extra, so more number of hours than in group 1 when they are being given lorazepam. So we can immediately visually see this. This is all about continuous and numerical data. So now what if we have a categorical variables, this variable where we want to find out frequencies of the, say, the outcome. So I will just explain this in this data set itself. So if you have a categorical variable like this or a nominal variable, so group is of course a nominal variable, we know. Just put this into the variables box. Since this group has been coded as 1 and 2, so 1 is lorazepam, 2 is this has been coded as 1 and 2. It is falsely showing us means and medians. So we don't want this. We in fact want percentages. So what I wanted to do is go into tables, which is the last drop down option. Go into tables and select frequency tables. So frequency tables will actually give you your proportions and percentages and frequencies. So you can see here. So group 1 and group 2, you can see how many data points there were, what is the percentage, so on and so forth. So you can see that in both groups there were 10 participants. It's obvious because these are the same participants, week 1, then week 2 and week 3, and 50%. So 50% 1 and 2. So this is quite obvious. But if you had another category variable, you would see proper frequencies of it occurring, the percentages. If you have missing data, then it will show you the valid percentage. It will exclude the missing data, and it will give you a cumulative percentage. So this is how you perform a descriptive data analysis in JAS. Now, does anybody have question? Oh, before I forget, if you want to plot pie charts, which would give you a visual idea of your categorical variables, just go into basic plots again, where we selected your histogram or distribution plot, and select pie charts. It's as simple as that. So I hope you can see why this is a very easy to use software, even easier than SPSS or Stata. Right. You can see a pie chart, 50-50. So right. Okay. So before I proceed, does anybody have any question or any doubt, or is anybody stuck at any stage? Or if anybody has something to say or something like that, you can put it in the chat box. So while you are doing that, it is almost 9:30. We have another hour, which is perfect. We would touch upon how to perform a chi-square test, a t-test, and an ANOVA and correlation. So we would touch upon all these things. First, we'll do the t-test. I will show you how to perform a t-test, and from there on, I will stop my screen presentation, and I would want participants to show the screen and do it, and I will guide you through it. One more thing which I would like to add, I have not explained how to import data into JAS. So you cannot import an Excel sheet into JAS. You would need to convert an Excel sheet into a CSV format, also known as a comma separated values format. So how to do that? I've shown it on my YouTube channel, Stat. You can go to the core concepts playlist, and I've shown how to do that. It's easy. If you open an Excel file, top right part of the screen, click on Save As, and save as a CSV file or a comma separated values file. And then go here, you can open from your computer and open any recent files you have. So that is how you do it. Okay. Mind you, in your data sheet, your first row should be variable names. Always. Your first row should be variable names, and after your first row, everything should be your data. There should be no stray values, no missing rows, no missing columns, so on and so forth. So that's important data into JAS. So that is known as data cleaning. So how you clean data. So I've explained some of that in on my YouTube channel. Okay, so now the second, I would want you again to go into this top right part of the screen, your 3 stacked horizontal bars. Go into open again data library, and this time I want you to open t-tests. And I want you to open this directed reading activities. Again, don't open this green icon. Don't open this JAS file. Open this table, the data file. Not the JAS icon, the table icon. Since I am short of RAM, I'll close this JAS file. Let me close the presentation as well. Okay. So in the meanwhile, does anybody of you have any doubt again? What is better, JAS or Power BI? I'm not sure about Power BI. I am guessing Power BI is a software for sample size calculation and power. If that is the case, then Power BI might be better because this power module in JAS has just recently been inculcated into JAS. So I am not aware about Power BI, but the purposes of learning data analysis, for the purposes of learning how to perform data analysis, and for the initiates, JAS, there's no tool like JAS. JAS trumps all other softwares because the sheer user interface and simplicity. Definitely JAS for data analysis, for analysis. All right. So I hope you all have opened this directed reading activities data set. Okay. So as usual, I will explain what this data set is. This data set is a data set on a reading test. So this test has scores. The students have been given, have given this test and they have been graded. So this DRP column, so ID column stands for the ID of the children. This should be a nominal variable. This DRP stands for the scores. So the test name is some directed reading power test, apparently. So that is why it has been abbreviated to DRP. So this is a test score. So this is a numerical or continuous variable. So and among these 44 children, 21 children have been primed for the test. They have been given certain exercises, certain training for the test. So we are denoting this to be the treat group. So these have been so-called treated for the test, primed for the test. And the control group, they have not been coached, they have not been trained for the test. What is the difference between biostatistics and typical statistics? It is me. Yes. So yeah, So it is just the application of statistics in the field of biology. So usually statisticians are are more into the mathematical side, and they deal overall with like different fields. And biostatisticians typically mostly only deal with biology. So yeah. Okay. So again, just to repeat myself, the trade group of students have been given training for this DRP test, and the control group have not been given any type of prior training. And we are now setting out to see whether the training has had any effect on the test or not. So we want to see, yeah, methods are the same. Methods are usually the same. Biostatistics usually has a few unique type of tests like Kaplan survival analysis, Kaplan-Meier, Cox proportion analysis, stuff like that. So which are more to do with your survival rates or outcome rates. That so what yeah, methods are mostly the same. I mean, you can apply all of these concepts to other fields as well. You can apply the same concept which I'm talking about to business. You can apply the same concepts to geology. So the same concepts hold true for all the sciences. But yes, method is the same. Okay, so the so before performing any type of test, we would like to formulate our null hypothesis for this. So our null hypothesis is that is a statement of no difference. Remember, so our null hypothesis is that there exists no difference in DRP test scores between the treated group and the control group. So statement of no difference, there exists no difference in the DRP score between the treatment group and the control group. And a two-tailed alternative hypothesis would be there exists a difference between DRP in the DRP scores between the treatment group and control group. So if you have listened closely, I've said a two-tailed alternative hypothesis. So we are not commenting whether the treatment group have higher scores. We are just saying that there is a difference. If this was a one-tailed alternative hypothesis, we would say that yes, the treatment group has a higher, there is a difference, and the treatment group has higher scores. That would be a one-tailed alternative hypothesis. But for the most part, for observational studies, it is better to use a two-tailed alternative hypothesis. One-tailed alternative hypotheses are usually used in clinical trials. Usually. So that is how we have formulated a null hypothesis before actually performing any type of data analysis. So this is very important, and I encourage all of you before performing any type of statistical analysis, any type of analysis to clearly formulate a null hypothesis, because this will help you when you're interpreting your results. It's very important. Otherwise, you leave a higher chance of misinterpreting your results. So please always formulate a null hypothesis before setting out to do an analysis. Right. Now, I hope all of you have taken a screenshot of which statistical test to use for what type of data. So if you can just quickly refer back to your screenshot. Here, the dependent variable, remember a dependent variable is the variable which is the outcome variable, or which we are measuring. So the outcome variable of interest. So the dependent variable is your DRP test scores, and your independent variable is the variable which affects your dependent variable. So in this case, this type of training, we are thinking that this might affect DRP scores. So this group would be your independent variable. Just this G column just is a coded column for your group. So you can disregard this G column. So again, dependent variable is continuous DRP, independent variable is categorical group. If you refer to your screenshot, you would see that when your dependent variable is continuous, and your independent variable is categorical, you can use either a t-test, ANOVA, or linear regression. Also, if you remember, a t-test is used only if we have only two groups. Here we only have two groups, right? Treatment and control. We don't have a third group. And ANOVA, or an analysis of variance, is used when you have more than two groups or three or more groups. Here we have only two groups. We are fine to use a t-test. Now, we are not using a regression because there are more assumptions in the regression for a test, and it is a bit more complicated procedure to explain. So t-test is fine for the purpose of this exam. So how to perform a t-test? So just select this t-test drop down sort of option here, and here you will see classical and Bayesian. So classical, also known as frequentist statistics, is a type of philosophy of statistics, and Bayesian, which was initiated by Thomas Bayes, is a type of is a different type of philosophy. So Bayesian statistics are not that commonly used in medicine, in medical science at least. They are more used in machine learning and meta-analysis and network meta, so on and so forth. But and it is a bit more complicated to infer the results of Bayesian statistics. For the purpose of you would also, most of the papers you see or most of the analysis you will be doing will be classical or frequentist type of analysis. So you won't do the Bayesian analysis. So for the remainder of this workshop, I would only touch upon a classical statistics, not Bayesian. Now, underneath the classical statistics option, you have your independent sample t-test, paired sample, and one sample. So what is the difference between these three types? So one sample t-test is when you have only one group, and the second group you have knowledge about the mean and the standard deviation of that group. So here we have clearly we have two groups, the treat and control group. Now, paired samples t-test. So we would use a paired samples t-test when the same group of participants are being measured before and after something. So in the first example of your sedative drugs, if you remember your sleep sedative drugs, the number of extra slept, those were the same 10 patients. So 10 patients, week one, they were given drug one, and the same 10 patients after a certain period of time, they were given drug two. So here, if we wanted to compare the mean number of hours slept between these two groups, we would use the paired samples t-test, because it's a paired data, right? Same group of patients paired before and after. But in this case, all of these 44 participants are unique. So these are not the same children. So these are independent or unpaired. So we would use the independent samples t-test, also known as the unpaired samples t-test. So just select this independent samples t-test, and you will be shown this screen. Now, as I've mentioned, before performing any type of statistical test, it is always desirable to perform an exploratory data analysis, descriptive analysis. But due to constraint of time, I won't do a descriptive analysis for this data because I've already shown on one data set. So if you want to perform descriptive analysis, just follow the steps I have shown in the previous video. Just go to descriptors and perform your analysis. All right. So now, how do we perform a t-test in JASP? So it is very simple. You can see that the dependent variables box is right here and grouping variable box is right. So just drag and drop your dependent variable, that is your test score. Remember, your test score is your dependent outcome variable, and your group is your grouping variable or independent variable. This is very easy. And voila, you have your p-value, you have your t-statistic, you have your degrees of freedom. You have everything right here. Now, before interpreting the results of this test, it is important to check assumptions, whether these have been fulfilled. So what are assumptions? Assumptions are certain assumptions of the data which need to be fulfilled for a test to work properly. So test might give you all results, but these assumptions need to be checked for these results to be valid. So what are the assumptions of t-test? How do you know what assumptions? You can either Google assumption of t-test, but that's tedious, right? So JAS very conveniently has taken care of this and given, if you see this small eye, blue colored eye, if you hover around that, on that, you would see this show info for this analysis. If you select that, you'll see all the assumptions for t-test right in front of you. So it's very convenient. So these are the 4 assumptions for t-test. So your dependent variable is continuous, and that is correct. That is fulfilled. Your dependent variable is continuous. The observations in both groups are a random sample of the population. Now, this has to do with statistical methodology and not with any type of formal analysis. So we hope that the researchers have randomly sampled the children. The third assumption is that the dependent variable, the DRP, your score, dependent variable, is normally distributed in both populations. So what is normal distribution? If you have seen a nice smooth bell-shaped curve, that is what I mean normal distribution. That is when your measure the central tendency, your mean, median, mode align at the exact same point. That is a normally distributed distribution. So how do you check distribution of data? How do you check for normality of data? So you can do it by 2 techniques. You can do it by visualization techniques, or you can do it using statistical test. So visualization techniques, you can use histograms, which I've already described in descriptive statistics. You can visualize if your histogram and your density curve is nicely normally distributed in a bell-shaped fashion. Or the second type of visualization is known as QQ plot. I won't touch into what QQ plots are and how you infer them, but that can be used. You can also use a statistical test. So the Shapiro will test for normality, and that is the easiest to use, and I will show you how to do that. Just if you scroll down here, you will see that you will see some assumption checks: normality and equality of variances. So for normality, just select this checkbox, easy peasy, and you will get some p-values here. If you read this note, significant results, that is a p-value, significant p-value less than 0.05, suggests a deviation of normality. So if you have a p-value which is less than 0.05 here, any one of these, mind you, need not necessarily be true that both of these p-values need to be significant. If any one of these p-values is significant, that means the data are not normally distributed in one of these groups. And in this case, these results would be invalid. We would need to use a non-parametric test, which would be this man-Whitney test. So if you select the checkbox, if this assumption is violated, then you need to use a non-parametric test. The man-Whitney test is non-parametric test. Select the checkbox, you'll get a p-value here, and use this p-value, not the student's t-test p-value. Okay, but here both of these p-values are non-significant. That means your data are normally distributed. It's very nice, and you don't need to use a non-parametric test. What was the fourth assumption? That's the assumption of homogeneity of variances. So the population variances in both groups are homogeneous or equal. How to check for that? Very easy. Just select this checkbox underneath the normality assumption option, and the same concept applies here. If the p-value is less than 0.05, is the p-value significant? That means that the assumption is violated. The variance of data are not homogeneous. And if the p-value is not significant, like in this case, it is more than 0.05, that means that the variances are equal or homogeneous. So that is good. We want homogeneous variances. If supposing that this p-value was non-significant. Suppose it was significant. Suppose there was no homogeneity of variances, then we would use a Welch t-test here. But this p-value is non-significant. Data are homogeneously, the variances are homogeneous. We are good just to keep your students t-test. We are good with that. Okay. So an easy way to remember N normality violated, then use M, man Whitney. So NM and variances V violated, VW Welch. So NM VW. It's easy. It's easy to remember that. All right. So now you see that the p-value is 0.029, 0.03, which is less than 0.05, your p-value threshold. That means that your result is significant, that there exists a difference. So you will reject your null hypothesis. So when your p-value is significant, you reject your null hypothesis and you accept an alternative hypothesis. So you reject the statement of no difference. You reject that there is no difference between these two groups. So you accept that there is a difference between these two groups. So how do we know where the difference is and which group has a higher score? We can visualize that thing. You can go to descriptive plots right here, which will give you your mean, which is 95% confidence interval for that mean. And you can actually see that this plot render, and can actually see that the students who had been given training on that for that test or treatment group actually scored higher. Their average score was actually higher than your control group. And if you select this descriptive checkbox, you will get descriptive statistics right here. So you will get your mean. You can see that the students who had been given training scored on average 10 points higher than students who had not been given the training. You can see that your dispersion of data right here, standard deviation, standard error, coefficient weight, for all that. And if you want a different type of plot, bar plot, just let us bar plot. You will get a bar plot like this with confidence intervals. This denotes the mean, of course. And if you want a very fancy type of plot known as rainbow plots, which will give you raw data points, box plots, and distribution curves. Yeah. So these are known as rainbow plots. So it is called that way because it looks like this is rain falling. So actually, this is better seen in horizontal display. So this will actually give you how your raw data points look like, your box plots. Remember that box plots are your medians with an interquartile range. So yeah, so you can see that these are clouds and this is rain falling. So that is why it is known as rain cloud plots. So basically, this is your box plot, your median, your interquartile range. And you can see that the median for the treatment group is higher. This is of course exactly the same thing that we saw right here. I won't touch upon effect size for this workshop. It is best left for another workshop. Location parameter just gives you your mean difference. So we saw that the mean difference was 10 right here, almost approximately 10, 9 point something. And if you select this location parameter, it will give you the same mean difference in this table. I'm not sure why it is taking so much time. It should not take so much time to show the mean difference. Yeah, it's loading now. But it will show you the mean difference with 95% confidence intervals. So yeah, 9.9. So this is exactly the same thing, the mean difference. Okay, so that is all about t-tests. Now, now I would want someone to share the screen. I hope it is Okay. Would someone voluntarily like to share the screen? You can put in the chat box. You can say that I'd like to share my screen.
Yeah, you can put it in the chat box and I'll give you permission to share your screen.
Yeah, please. Okay, Nidhi, great. Sarah, next time, the next data set, I think you can share your screen. Right now, let Nidhi share her screen. And for chi-squared, so Nidhi would be doing chi-squared and ANOVA, and Sarah, you would be doing chi-squared. So I'll stop my screen sharing. I think that would hog a bit of bandwidth. All right.
Nidhi, I've given you, yes.
Okay. So I want you to go to, yeah, show main menu. Open. ANOVA, please go to ANOVA. All right. So just scroll down a bit. Some more. Yeah, the pain thresholds. Yeah, the last one. Yeah, the pain thresholds. Yeah, open the data. Yeah, perfect. So I hope everyone has opened the pain thresholds data set. All right, now let me give a description of this data set. This is a disclaimer. I haven't made this data set, so if anyone is offended by this, I'm sorry. Just take it in a lighthearted fashion. So this actually is a data set which measures pain tolerance in participants with different hair colors. So the hypothesis is whether patients with different hair colors have different pain tolerance levels. So if anyone is uncomfortable with the data set, please tell me. We can switch to a different data set. So okay. So the rightmost column, pain tolerance, denotes a measure in numbers of pain tolerance. The middle hair color column is the categorical variable or your grouping variable. So light blondes, dark blondes, light brunettes, dark brunettes. So so Nidhi, would you like to formulate a null hypothesis? So would you first tell me what a dependent variable and independent variable is here?
Hair color would be independent variable and pain tolerance would be dependent.
Perfect. Yeah, correct. And if you have the screenshot for the type of statistical test to use near you, would you like to? Yeah. Okay. Which one would you like to use here? Yeah, dependent is a continuous variable and your independent is a categorical variable. And you have more than two groups. So an ANOVA. So t-test you would use when you have only two groups. An ANOVA you would analysis of variance would use when you have more than two groups. So you have four groups: light blondes, dark blondes, light brunettes, dark brunettes. So you would use an ANOVA. Perfect. ANOVA, classical. Yeah, the ANOVA is fine. Let me explain what the all of these mean. So repeated measures ANOVA is like paired samples data. So if you have repeated measures, so if you have say measures of participants at baseline, then after say 1 year, 2 years, 3 years, then you would use a repeated measures ANOVA. Participants are the same. The same 10 participants you are measuring over one, say at 0, 1, 2, 3 years, then you would use a repeated measures ANOVA. And ANCOVA is known as an analysis of covariance. So when you want to account for a certain factor which might affect the dependent variable as well, so you would use an ANCOVA. And MANOVA is a multivariate analysis of variance when you have two dependent variables or more than or equal to two dependent variables. Then we will use a MANOVA. But let's keep it very simple. Let's use an ANOVA. Perfect. Right. So I won't say anything. I'd like to see how you approach performance analysis. Yeah. That you are going to 6 times. Yeah. Perfect. Perfect. Okay. That's good. So before going anywhere else, I would like to all of you to focus your attention on the table. So you can see just focus your attention on the p-value. The p-value is less than 0.05. Oh, I forgot. So can you please state your null hypothesis for this data?
There is no difference in tolerance with change in hair color.
Perfect. Okay, great. All right. So now the p-value is significant, but before, so is this result valid? So we need to check assumptions for our data for ANOVA. So yeah, all right. So the first assumption, residuals are normally distributed for each group. So I won't go into what residuals are, but we can check that assumption. So if you see the second drop down option, like here underneath model, you'll see assumption checks. The second, yeah, there below model assumption, yeah. If you select a QQ plots for residuals, you'll get a QQ plot. So this is also known as a quantile plot, QQ plot. And what you need to see is that the data points, the values, the dots approximately need to fall on a straight line. The straight line has been drawn for you, and these data points need to fall on the straight line. So if the data points are very, like they don't fall on straight line and they are very dispersed, then you would infer that your data are not normally distributed. In this case, you can see that your data points more or less are falling on the straight line, and that is why you can infer that yes, your data are normally distributed, your residuals are normally distributed. You can see that the tails there, the data points are not really falling on the line, but that is fine because the tails usually do not conform to a normal distribution. So that's fine. So so after looking at this, the data, all the points are falling on the straight line, more or less, and I think this looks fine. The data are normally distributed, so that's good. Okay, can we read the second assumption? If you go to the JASP icon, just down the task, yeah, okay. The independent variables are categorical, that is true. Undependent variables continuous. Perfect. So that is fulfilled. The fourth assumption, groups are independent. These, yes, of course, the groups are, so no group is repeated. So no two groups are the same. So no participants are same. All of them are different participants. So even the fourth assumption is fulfilled. The third assumption, again, is the same as t-test, homogeneity of variances. So how do we check for that? If you go again to assumption checks, there should be an option for checking the Brown-Forsyth test. The homogeneity test, the first checkbox. Yeah. So Nidhi, this is the same, you have to infer the p-value the same as how I explained to infer p-values in the for equivalence test, the homogeneity variance test and normality test. So can you infer what this p-value means in terms of homogeneity variances?
Now p-value is much higher, so it's more than 0.05. So that means it is not significant.
Yeah, it is not significant. So so now is this assumption met or violated? So are the data homogeneous or not?
No.
So the data are so so a significant p-value for this test. So the assumption test, so Shapiro-Wilk test or this homogeneity test, a significant p-value means that your I'll wrap this up soon in 20 minutes. So I think we can discuss ANOVA and chi-square and correlate. So a non-significant p-value means that data are homogeneous, and a significant p-value means that the data are not homogeneous. So this is completely opposite of the p-value you would infer for a normal statistical test. So this is opposite. So a p-value, a non-significant p-value here is desirable.
Okay.
So that is how. All right. So I think all of the 4 assumptions have been met, and this p-value, significant p-value, is a valid result. So this significant p-value means that we reject the null hypothesis. So we reject the statement of no difference, and we say that yes, there is a difference. But now there are 4 groups: light blonde, dark blonde, light brunette, dark brunette. So where exactly is the difference? So is there a difference between all 4 groups, 2 groups, 3 groups? Where's the difference? So we check that. For t-test, it is very simple, right? There are only 2 groups. We know that there's difference between 2 groups. For ANOVA, there are more than 2 groups, so 3 or more. So where is the difference? We can do that by doing what is known as a post hoc test. So if you go here exactly and you put this hair color into the other box, you would get a table soon. Yeah, here. So this will actually give you pairwise comparisons between groups. And if you just select this flag significant comparisons here, the checkbox left side of the screen, flag significant comparisons. Flag, last one. Yeah. And if you just select this, if you scroll this to the, scroll this to the, scroll the table to the leftmost point. Rightmost, so you can see that the starred p values are significant, less than 0.05. So you can see that between all of the groups, significant differences only lie between dark brunettes and light blondes, and light blondes and light brunettes. So there is a significance only between these 2 pairwise comparisons, and no other pairwise comparison is significant. And we can visualize that. So if you can please go to descriptive plots and hair color in the horizontal axis, and just select display error bars as well. It will take a little time. You can actually visualize. So if you just hover around on the graph a little bit, you can see the on the bottom right screen, you have those 3 lines, diagonal lines that you can, okay. It will, so we could actually see that there is a significant difference between light blondes and light brunettes. So we can actually see that as well. And what is the second one? I forgot. Second, if you scroll down a bit. Dark brunettes and light blondes. So between these, and you can visualize this as well. Okay. So if you again scroll down just a bit, tad bit again. So you see the P-Tukey. So this is a type of correction of p-value. So when you are performing multiple statistical tests on the same data, you need to perform you need to perform a p-value adjustment. So this is again a concept which we would not touch during this session, but later. So if you go, please go to assumption checks again, back. So now what will we do if either one of these fails? So what will we do if the assumption of normality fails? We would need to use a non-parametric test. So if you just scroll to the very end of this thing, very end, yeah, you can see non-parametrics. So here is a Kruskal-Wallis H test. So you can just put the hair color into this box and we'll give you a non-parametric alternative. So remember, for a t-test, it was the Mann-Whitney test, a non-parametric test. For ANOVA, it is a Kruskal-Wallis test. And here we see a p-value. Now, if you go, please go to assumption checks back again. Now, what will you do if the assumption of homogeneity is violated? What if you do is if the p-value is high? Then just select homogeneity correction, Brown-Forsyth or Welch, either of the two. And if you go to the first table now, the topmost table, you would get your correction, your Brown-Forsyth correction. So that is, so you will use this p-value if your assumption of homogeneity is violated. So, all right. So this is all about ANOVA. And okay, so Nidhi, thanks a lot. This was great and great job. Sarah, I believe you wanted to share a screen if you're still online. I think she left.
Yes,
okay. Yeah, would anyone else like to share the screen? And this will be a chi-square test, a very easy test. So, okay, so if no one wants to share a screen, I will share my screen and just show it to you. Chi-square and correlation. So, all right. So for the purpose of chi-square test, I want you all to go into show main menu, open data library, go to frequencies, and open dancing cats. Data set, not dancing cats and dogs, just a second one, dancing cats. And let me explain this data set. So in the meanwhile, you can post in the chat box if anyone of you had any doubts in t-test or ANOVA. Okay. So this data set measures whether cats learn to dance or not. They were trained to learn to do dance, and they were given two types of training. So after their training to learn to dance, they were given either food or they were just given affection, just petted. So you can see that 38 cats were given food after training, and all the remaining 162 cats were given just affection, just pets, no food. So that's kind of cruel, right? But this is a fictional data set, not so there was no animal cruelty involved here. So, and this column in dance indicates whether they learn to dance or not. So yes means that yes, they learn to dance, and no means that no, they did not learn to dance. So here your dependent variable is whether they learn to dance or not, and independent variable is the type of training. So your outcome variable is whether dance or not, and independent variable is type of training. So both of these are categorical or nominal. And if you see your screenshot, you would see that when your dependent variable is categorical and your independent variable is also categorical, you can use either a chi-square test or a Fisher's exact test or a logistic regression. So, all right. So to perform a chi-square test or a Fisher's exact test, go to frequencies. Underneath the classical option, go to contingency tables. Frequencies, the classical statistics option, and contingency tables. All right. And now here you can actually put your variables into rows and columns. So personally, I like to put my dependent variable into columns and independent variable into rows, but there's no such order or rule. You can do anything, but it is easier to interpret if you do like so. And just for interpretation purposes and yeah, I would like to change the column order. So I would like the yes to be on my left and food to be the top. So you can do so just by going to this options drop down menu and check your descending, and I'll give you this. All right, so you might see that I am getting this exponent function. So it is 10 raised to minus 7. So that is because you might be getting less than 0.001. That is because I have selected to get exact p-values from this preferences. All right, so now this might be looking more like your screen, so less than 0.001. Okay, so so what are the assumptions of chi-square test? There are very few assumptions. The first assumption is that these numbers should be independent. So the same cat should not be in both of these rows. The second assumption is that your total sample size should be more than 50. So that's fine. It's 200 is more than 50. Your third assumption is that no value in each of these 4 cells should be less than 5. If any of these values is less than 5, you would use a Fisher's exact test. So none of these values is less than 5, so we are good to go, and we can use a chi-square test. So that's not a problem. Now, we want percentages because it will increase our interpretability. So you can go to this cells option and you can select either row or column. So if you select row, it will give you your 28 out of 38, this percentage within your row. If you select column, it will give you percentage within that column. It is preferable to only select 1% because selecting more than 1 will increase confusion. So just select within row if you have your dependent variable in your column. And you can see that 73% of cats who were given food as a reward learned to dance, whereas only 30% cats who were given affection as a reward learned to dance. So you can see that food is a better motivator to learn to dance than affection. Okay, and the p-value is significant. So that means that you reject your null hypothesis that there is no difference between the type of training on the cat's ability to learn to dance. So there is a difference in the type of training, and we can see that food as a reward, a higher number of cats learn to dance. All right, so if you go to statistics, you can actually perform a Fisher's exact test by selecting odds ratio. So if you have any value in any of these cells, you would want to perform Fisher's exact test. Just select odds ratio, and this will perform Fisher's exact test for you. So again, the result corroborates with your chi-square test. So p-value is significantly less than 0.001, and that's great. So I am just running through this chi-square test. 5 more minutes. Yeah. So chi-square test, I think this was pretty straightforward. I would want you all to open a last final data. So just go to open, go to data library, go to regression, and go to album sales, the second one. Okay, so if any one of you has any doubt, you can reach out to me on LinkedIn. You can also comment on any of my YouTube videos. I have, so stat is my YouTube name. This Suhrud Panchawagh is my LinkedIn handle, and this is my email. Oh, I have sent it on personal chat. Do you think JASP is better than SPSS? So in terms of being open source, JASP is definitely better because it's free. SPSS is paid, but in terms of functionality, SPSS is better. JASP is user friendly, and anyone who is setting out, especially non-statisticians who are setting out to do data analysis, I 101% recommend them to start learning from JASP, not SPSS. Okay, so I hope all of you have opened this data set, and here you can see all of these variables. Basically, we want to see whether, so this is a data set of a certain music band, and you can see that we want to see whether there is an association between the number of advertisements of the band and the sales. So we want to see if increasing the advertisements increases the sales of the band. Let's not concern ourselves with these two variables. That'll take a little longer. So we want to see whether advertisement and sales is associated. So sales will be a dependent variable or an outcome variable, and advertisements are independent variable affecting your dependent variable. If you see your screenshot, dependent variable is continuous and the independent variable is also continuous. You do use either correlation or linear regression. Here I won't go into regression, just correlation. To do that, go into regression, this drop down menu, go into correlation, and put both your independent and dependent variable into your variables box. I'll let this load first. Yeah. Okay, so there are certain assumptions for correlation. I won't go into these, but the assumption of normality is very important. So how do you check for assumption? So you can go to assumption checks here and select pairwise normality. So if the p-value is significant, that means there is no normality and data not normal. The p-value is significant, that is a bad thing. So we can't use a parametric test. We can't use a Pearson's R. We need to use a non-parametric test. So in the case of correlation, Spearman's rho or Kendall's tau both are non-parametric alternatives to your Pearson's correlation. So because your Shapiro test, the p-value is significant, data not normally distributed, I'm using a non-parametric test, Spearman's test correlation, and you are using this. And your p-value is significant. That means that there is a significant association between these two variables. We reject the null hypothesis that there is no association between adverts and sales, and we accept the alternative hypothesis that yes, there is an association between these. And you can see the association. You can see the scatter plot. You can plot this, and you can actually see a line and data points. You can see that this load first. Yeah, so you can see that there. So as there exists a positive. So as advertisements have increased, so adverts is on the y-axis and sales are on the x-axis. So as adverts increase, sales also increase. So there's a positive association. So a positive Spearman's rho correlation coefficient indicates that there's a positive association. As one variable increases, the other also increases. If this is negative, that means as one variable increases, the other decreases. So this is positive, so both are positively related. Okay, so I think I'll stop my screen sharing and I'll open this chat box or channel to any doubts.
Yeah, we have a little time, so 1 or 2 questions would suffice. So if you have any questions, you can either message in the chat. Okay, we have one question.
Yeah, how can it? Yeah, excellent question. So if you in JAS there is an option. Okay, let me again start screen sharing. So here there is an option. So you can either select a small downward pointing arrow and copy all of this and paste this into a Word file, or even if you want to just copy one table and not the entire thing, just one table copy. If you select export, it won't give an option to export into PDF, into Word. It will give you an option to export into PDF or LaTeX. So if you want LaTeX, you can export into LaTeX or whatever. Or HTML, I'm sorry, not LaTeX. LaTex you can export your copy LaTex. Okay, any other doubts? Yeah.
Any other question, guys?
So any books or suggestions? Okay. So mine was a sort of a unique journey, and I actually learned using data sets from hospitals. So not even example data sets, and I learned it from a PhD biostatistician from Johns Hopkins. So I was fortunate enough, but I know that it is difficult for people with a medical background, a biology background to learn statistics. So that's why I have started a YouTube channel where I aim to practically explain, because I'm aware that people from biology background don't have enough time to read mathematics and statistics. So in my YouTube channel, I aim to do exactly that. I aim to teach biostatistics in a very practical manner. So if you just hold on a second, I will share my YouTube channel link. And I think after the if there's a post session survey of how you like the session or something like that, perhaps Simply Neuroscience can also share the link to my YouTube channel, perhaps in a link post or something like that.
Yeah, sure. Yeah.
Yeah. Oh, into our phone or tab. Yeah. So actually, JAS. If you go to the JAS web page, there's an option to run JAS in your browser, and if you go there, it'll take you to a third-party link. It's called Rollup. You have to make an account in Rollup, and if you do that, you can access JAS uh, through your browser, on your phone, on your tab. And you can perform data analysis on your phone as well through the browser. So it's very convenient that way. But it is not, if you have a big tab, then that's great. But on your phone, it might be tricky to navigate through the sections. So yeah, you can do it on a phone, but not the ideal way. Okay. So yeah, I have shared my YouTube channel link in the chat box. So you can go to my YouTube channel and check out my videos. And yeah.
All right, great. So looks like time is up. I hope you all enjoyed and learned a lot from this session. And thank you so much to Suhrud as well for giving an amazing presentation. And thank you all for being an amazing audience. So I will stop the recording now.
Yeah, so I've also shared so this is www.requirestats.in. So this is